Welcome to the MagBio HMW DNA Kit protocol. The HighPrep HMW DNA Kit is engineered to streamline the process of isolating high molecular genomic DNA. It's capable of extracting DNA from various sources, including saliva, cultured cells, tissue samples, bacteria, and whole blood. This DNA extraction protocol guarantees high molecular weight, 50 to 300 kb plus DNA with exceptional purity. This makes it perfect for demanding applications such as long-read sequencing and genotyping. Now, before we get started with the protocol, let's prepare our reagents. Note that there are 2 reagents that require reconstitution using 100% ethanol. These reagents are the HMW1 and HMW2 buffers. First, let's add 100% ethanol to the HMW1 following the measures on the table. Now, mix the solution by inverting the bottle for a few seconds. Now, do the same process of adding 100% ethanol, but this time to the HMW2 buffer. Now, mix the solution by inverting the bottle for a few seconds. For our protocol, we have chosen to run it on a blood sample. Here are a few tips when performing the protocol on a whole blood sample. Now that you've prepared your reagents, let's get started with the protocol. For step 1, add 20 microliters of the Pro-K solution to a 1.5 milliliters tube. Add 100 microliters of the NB elution buffer. Then add 200 microliters of whole blood. And then add 300 microliters of HAS buffer. Pulse vortex for 1 second or invert the tube 5 to 10 times to mix the sample, then incubate the sample at 55 degrees Celsius in a thermomixer for 20 minutes. If using a water bath, then invert tube 3 to 5 times during the 20 minutes incubation time. To assess the lysis performance accurately, make sure the sample is showing a greenish-black color. Next, bring the sample to room temperature and add 5 microliters of RNase A. Incubate for 3 minutes. Then, grab the MAG HM1 particles tube and pulse vortex for 1 second for 5 times before adding 10 microliters of MAG HM1 particles to the sample. Add 430 microliters of 100% ethanol pulse vortex for 1 second, or invert the tube 5 to 10 times to mix. Now place on a mini rotator and rotate the samples at room temperature for 10 minutes. Note that it is normal for lumps of beads and DNA to form during rotation, so do not break the lumps. Briefly spin tube on a mini centrifuge for 2 seconds. Place the sample on a magnetic separation device for 5 minutes, or until the beads are completely cleared from the solution. Note that you should discard the supernatant by pipetting the liquid out. Make sure to remove any residual liquid from the tube cap. Now add 500 microliters of the HMW1 buffer. Pulse vortex the sample for 1 second or invert for 10 times to mix. Once done, put the sample on the mini rotator and gently rotate the sample at room temperature for 5 minutes. Briefly spin tube on a mini centrifuge for 2 seconds. Place the sample on a magnetic separation device for 5 minutes, or until the beads are completely cleared from the solution. Note that you should discard the supernatant by pipetting the liquid out. And it's important to make sure to remove any residual liquid from the tube cap. Repeat the HMW1 wash for a second time before proceeding. Then, now you can add in the same way the HMW2 buffer to the solution. Pulse vortex the sample for 1 second or invert for 10 times to mix. Once done, put the sample on the mini rotator and gently rotate the sample at room temperature for 5 minutes. Briefly spin tube on a mini centrifuge for 2 seconds.
Place the sample on a magnetic separation device for 5 minutes, or until the beads are completely cleared.
Discard the supernatant by pipetting the liquid out, and make sure to remove any residual liquid from the tube cap. Repeat the HMW2 wash for a second time before proceeding. Then air dry the magnetic beads at room temperature for 5 minutes. Add 50 to 100 microliters of MB elution buffer to the sample. Do not use a regular pipette or vortex to mix the sample. Instead, use a pipette with wide bore tips. Once done, do as follows. First, incubate the sample at 55 degrees Celsius in a thermomixer at 500 revolutions per minute for 5 minutes. Remove the sample from the 55 degrees Celsius incubation and gently mix the sample by pulse vortexing for 1 second. Incubate the sample at 55 degrees Celsius in a thermomixer for a second time at 500 revolutions per minute for 5 minutes. Place the sample on the magnetic separation device for 5 minutes, or until the MAG HM1 particles are cleared from the MB elution buffer. On the final step, transfer the eluate, cleared supernatant, to new 1.5 milliliters microcentrifuge tube and keep at 4 degrees Celsius or for subsequent applications. Note that for long-term storage, you should keep the DNA at minus 20 degrees Celsius. Do not freeze and thaw repeatedly. And that's how you run a HMW DNA Kit protocol.